Learning outcomes. After studying this module, you shall be able to know what is fiber, what is the classification of fiber, how will you identify whether a thread-like structure is a fiber or not, which type of methods are used in examination of fiber, what is the chemical examination of fiber. Now let us learn about the fibers. Fiber is fine thread-like piece as of cotton, jute or asbestos. In forensic science, many forensic scientists use fibers as a means of determining the nature of the item worn and in some instances can be used as precise as to identify the maker of the garment and thus the manufacturer. In some instances, this technique is so successful that garments that are rare or indeed specially made can be identified and thus a list of possible suspects drawn up simply by number of units sold. Fibers obtained from natural sources have been used for production of textile and other products for thousands of years. Carpets, tents, sails, ropes and cordages can be traced back over 3000 years. More recently, textiles have found wider application and may be designated as architecture. Just like hair, fiber are also considered as trace evidence. In many cases, identification or matching of fiber evidence is essential in solving a crime. Criminalists examine size of fiber, its crime, color and cluster. They may also examine its cross-section, debris sticking to it and even soil damage. The determination of reflective index of fiber can also be done so as to allow for matching and comparison with existing databases. Chemical testing may also be employed for identification purpose. Fibers can rarely be used to make an exact positive identification unless torn fibers can be physically matched at their edges. A side-by-side comparison of a standard sample and the crime scene sample is generally conducted first to compare color and diameter of the fibers. Morphological features that may aid comparison include striations depicting on the fiber structure, pigmentation and the shape of the fiber through a cross-sectional view. Standard samples should be taken from the crime scene for later comparison with suspect samples. Using microspectrometry, light wavelengths are passed through the fiber, some of which are absorbed and some of which pass through. By examining which wavelengths are absorbed, the nature and the color of the fiber can be determined. Gas chromatography is used to study the chemical composition of the fibers by decomposing the fibers to gas and analyzing the individual components. However, this is a destructive method which should only be employed following non-destructive methods.
Structure of fibers. Fibers vary a great deal in terms of size, with a range varying from 10 to 50 micrometers. 1 micrometer is equal to 1 millionth of a meter. 1 millimeter is 1000 microns long. In comparison, a human hair is about 70 microns in diameter, and fine talcum powder is about 10 microns in diameter. In general, the softer materials will have the smallest fibers, and the more durable materials will have the largest fibers. The cross-section or the transverse section of a fiber varies according to type. Natural fibers, as they have their origin in plants and animals, will look rough on the edges, with a rather rough shape to the cross-section as well. Synthetic fibers, on the other hand, will have a greater variation in the cross section. That is, they may be round, oval, bilobal, that is, having two lobes, and trilobal. The color of a fiber may be natural to the fiber, or it may be added later, that is, bleached, dyed, or printed. For example, cotton is naturally white, but cotton is often dyed. When a natural fiber is dyed, the color will permeate the fiber. In synthetic fibers, the color can be seen as tiny specks of pigment within the fiber itself. Next, moving on to classification of fibers. Fiber has commonly been classified into two categories, that is, natural and man-made fibers. Natural fibers have been further classified into three categories, that is, plant, animal, and mineral fibers. Under plant category, we have cotton and flax. Under animal, we have wool, mohair, and silk. And under mineral, we have asbestos. Next, moving on to man-made fibers. These have been further divided into two categories, that is, regenerated or synthetic. Under regenerated, we have rayon, cellulose, protein, glass, and rubber. Under synthetic, we have polyamides, polyester, acrylic, polyolefin, and vinyl. Commonly found at all crime scenes, fibers will either be natural or man-made. They are generally circular in cross-section and have scalar cuticles. They tend to be readily transferable and often intertwine with other fibers, making them more persistent. The application of heat may cause them to curl. However, since the development of nylon in 1939, man-made fibers have increasingly replaced natural fibers in garments in particular. Most natural fibers have distinctive appearances that can be detected using microscopy. However, silk and synthetic fibers are produced by drawing out and solidifying a liquid, giving them a smooth and unfortunately indistinguishable surface. These fibers can display a variety of cross-sectional shapes, though rarely circular. They are almost always dyed and are less persistent on surfaces. Common examples of synthetic fibers are polyester, nylon, and rayon. Next, we move on to the identification and examination of fibers. The chemical instruments needed in the identification of fibers are very simple, and except for some of the chemical reagents used should be found in any chemical laboratory. The microscope need not be for very expensive type, but should be provided with lenses of good quality. It is very essential to record the result of microscopic examination for future references. First is visual examination. This is the simplest method of fiber identification. Single fibers and tufts may also be found during visual search of an item because of the transference of the textile material. Visual search can also be done by using a simple hand lens and is suitable in cases of burglary where the culprit has left fibers at the point of entry, on knives, or on clothing from a body which has been transported in an item, such as a carpet and following other type. Fibers are collected with the help of forceps and kept in a closed and labeled container for further examinations. The advantage of this approach is that the recovered fibers can be easily removed from the container for further examination without the need for searching amongst background debris. Various methods are employed in the collection of trace evidence. The method used depending on the type and nature of evidence. Larger items, such as long fibers, may be collected by hand or tweezers. One of the simplest methods of recovery is to shake the item over a sheet of paper or container. However, this does not allow for the exact location of evidence on the item to be documented. Some particles will not be dislodged by shaking the item. Therefore, brushing the item may be necessary. A common method of trace evidence collection is the taping technique, particularly beneficial in the case of fibers and hairs. A strip of clear adhesive tape is applied to the surface, peeled off, and placed against backing card. This allows a note to be made of the exact location of the trace evidence. Vacuum lifting is a particularly useful method of trace collection, as fibers are mostly transferred in the form of single tiny fragments rather than clumps or threads, and hence are not generally seen by the unaided eye. Now moving on to the microscopical examination. Microscopy is the essential element in almost all aspects of the forensic examination and comparison of fibers. On the most fundamental level, microscopes are necessary to see fibrous evidence no matter what observation or test are being performed. Thus, microscopical observation is used to detect fibers, to characterize them, and in most cases, to compare them to a suspect source. There are some common microscopes which are used in identification and examination of fibers. They are given as stereo microscope. This is used for low magnifications where its long working distance and upright image assist in searching small items of evidence or tapes and in recovery and manipulation of individual fibers. Next, moving on to polarizing microscope. The polarizing microscope is similar to a conventional biological microscope with additional features which facilitate observations and measurements with plain polarized light. It is an easy, quick, and non destructive technique to determine the generic polymer class of manufactured and synthetic textile fibers. Beyond the immediate characteristics used to discriminate between polymer types, the examination of fibers in polarized light provides valuable information about the production and finishing of the fiber after spinning. Next is comparison microscope. This instrument permits direct comparison of such fiber properties as morphology, diameter, and color. Fluorescence microscope. Many dyes used to color textiles have fluorescent components, and their response to certain wavelengths of light can be useful in comparing textile fibers. Not all textile dyes fluoresce, but fluorescence comparisons should be performed regardless. If the question and known fibers both fail to fluoresce, that is another point of meaningful comparison. Fluorescence occurs when a substance is excited by specific wavelengths of light. A light of relatively short wavelength illuminates a substance, and the substance absorbs and converts a certain small part of light. Most of the light is not absorbed by the substance, is re-emitted, which is called fluorescence. The fluorescent light has lost some of its energy, and wavelength will be longer than that of the light source. Certain dye combinations may produce fluorescence of a particular intensity and color, both of which should be noted during the examination. Fibers dyed with similar dyes should exhibit the same fluorescence characteristics, unless the fibers or dye have been degraded by UV exposure, bleaching, or some other similar means. It is important to consider these factors when collecting known samples. Now moving on to instrumental examination. Laboratory examination of fibers is principally performed using a polarizing light microscope. Using this instrument and oils of known refractive indices, a scientist can examine and compare color, thickness, cross-sectional shape, amount of delustrant, and composition. Other instrumental techniques, such as FTIR, are commonly used to determine synthetic fiber composition. UV visible microspectrophotometry can be used to discriminate color in the ultraviolet and visible regions that cannot be discerned visibly. Analysis of dyes present in fibers can also be performed. As is the case with glass and paint, a positive association in forensic fiber analysis is usually not a conclusive identification. A positive association will typically result in a conclusion that a questioned fiber is consistent with a known fabric. The importance of this finding may be directly related to how rare or how common these specimens may be in terms of availability. Again, no statistical data is typically applied to this type of conclusion. Under this, first we have infrared spectroscopy. Infrared spectroscopy is an important tool in determination of functional groups within a fiber. Functional groups present in the polymer absorb IR energy at wavelengths characteristic to that particular group, and thereby resulting in changes in the vibrational modes within the functional group. As a result of the infrared absorption characteristics of the fiber, specific functional groups can be identified. Infrared spectroscopy of fibers can be carried out on the finely divided fiber segments pressed in a salt pellet or through the use of reflectance techniques. Functional groups in dyes and finishes can also be detected by this technique. Next is ultraviolet visible spectroscopy. The UV spectra of fibers, dyes and finishes may offer clues regarding the structure of the materials. Besides, may also demonstrate the nature of electronic transitions that takes place within the material when light is absorbed at various wavelengths by unsaturated groups, giving an electronically excited molecule. The absorbed energy is either harmlessly dissipated as heat, fluorescence, or phosphorescence, or causes chemical reactions to occur that modify the chemical structure of the fiber. Ultraviolet visible spectra can be measured for a material either in solution or by reflectance. Reflectance spectra are particularly useful in color measurement and assessment of color differences in dyed and bleached fibers. Next is nuclear magnetic resonance spectroscopy. Nuclear magnetic resonance or NMR spectroscopy measures the relative magnitude and direction or moment of spin orientation of the nucleus of the individual atoms within a polymer from a fiber in solution in a high intensity magnetic field. The degree of shift of spins within the magnetic field and the signal splitting characteristics of individual atoms such as hydrogen or carbon within the molecule are dependent on the location and nature of the groups surrounding each atom. In this way, the average structure of long polymeric chains can be determined. Line width from NMR spectra also can provide information concerning the relationship of crystalline and amorphous areas within the polymer. Next is X-ray diffraction. X-rays diffracted from or reflected off crystalline or semi-crystalline polymeric materials will exhibit patterns associated to the crystalline and amorphous areas in the fiber. The shape and size of individual crystalline and amorphous sites in the fiber will be revealed in the geometry and sharpness of the X-ray diffraction pattern and will provide an insight internal structure of polymeric chains within a fiber. Next, moving on to thermal analysis. The chemical and physical changes within fibers may be examined by measuring changes in certain properties as minor samples of fiber are heated at a steady rate over a given temperature range in an inert atmosphere such as nitrogen. The four thermal characterization methods are: first, differential thermal analysis or DTA. Second, differential scanning calorimetry or DSC. Third, thermal gravimetric analysis or TGA. And lastly, thermal mechanical analysis or TMA. Now, moving on to the characteristic features of fiber under different conditions. We have samples ranging from cotton, coconut, wool, jute, silk, polyester, glass, and nylon. The various characteristic features given in the table of physical examination, specific gravity, burning examination, and the microscopic examination. Next, moving on to wool burn test. The wool burn test has been shown in the illustration, varying from time ranging from 2 seconds to 20 seconds. Now, moving on to chemical examination. The chemical properties of fibers comprises the effects of chemical agents including acids, bases, oxidizing agents, reducing agents, and biological agents such as molds and funguses on the fiber, and chemical changes induced within the fiber due to light and heat. Acids and bases cause hydrolytic attack of molecular chains within a fiber, whereas oxidizing and reducing agents will cause chemical attack of functional groups by oxidation, that is, removal of electrons, or reduction, that is, addition of electrons. Such chemical attack can change the fiber's structure and possibly cleave the molecular chains within the fiber. Next is a table that shows reaction on fiber with use of different types of reagent. The samples taken are cotton, coconut, wool, jute, silk, polyester, glass, and nylon, and the various types of reagent that has been recorded in this table are concentrated hydrochloric acid, concentrated sulfuric acid, concentrated nitric acid, concentrated sodium hydroxide, ethyl alcohol, carbon tetrachloride, and its effect on the litmus paper.
Now let us summarize what we have learned in this module. Fibers are considered as class evidence and also known trace evidence at crime scene. It can be identified and examined with the help of physical and chemical properties. Fibers can usually be divided into two main classes, which are natural and man-made fibers. All fibers which come from natural sources are cotton, wool, silk, etc., and which come from man-made or synthetic are nylon, rayon, polyester, polyvinyl, etc. Fiber examination consists of comparison of samples obtained from known and questioned sources to conclude whether they are being originated from the same source or not. Fiber identification and examination can be done by several methods including visual examination, microscopic examination, instrumental examination, and chemical methods. Microscopic examination of a fiber includes some important microscope such as stereoscopic comparison and fluorescent microscopic, etc., which can also help in easy examination of fiber. Same as microscopic examination, instrumental examination of fiber also include IR, that is infrared spectroscopy, UV visible spectroscopy, NMR, that is nuclear magnetic resonance, etc. Chemical examination of fiber includes effect on fiber after treating with some reagents such as some acids like hydrochloric acid, nitric acid, and sulfuric acid, and bases like sodium hydroxide, etc.